Okay, take two. Okay, so hello teacher and to everyone who is watching my presentation. My name is Nguyen Khang and I'm a student from the biotechnology department of IU in Vietnam. So today I'm making the video to share about my own concern that I got after my class and my laboratory works. So let's begin now. Uh, my concern is about how do we know what amino acids look like in the protein structure. For example, we learned that there are two amino acids which causes sickle cell anemia. How did we learn that? And how do we know which amino acid that is? We don't really see the letters C, H, S, or N when we look through the microscope at the proteins. So, how did we get so advanced to understand the protein interactions? Um, my own concern sound kind of silly, right? But I think it somehow really necessary and practical to every student that don't have much chances to get in the laboratory and test out all the knowledge that they have learned. And me too, it's same with me. So that's what I concern now. So no more wasting your time. Now let's get it on. As I have studied, each amino acid has the unique property to tell apart and we have our own technique to remove those amino acids out of it out of the protein sequence. Therefore, by combining those two, we can tell that it is possible to determine the protein sequences. So the question is that now, what is the protein sequencing? Yeah, the protein sequencing in the definition is the practical process of determining the amino acid sequence of all or part of a protein or peptide. Uh, to now, there are many there are many methods to use, and the methods are widely used now are the methods belonging to the mass spectrometry. So, but before we get in too deep in that, I found an interesting fact about this protein. That is uh, about the very first sequencing determination. Mr. Frederick Sanger, who is I found that he the first one to determine the very first protein sequences, and it's more than a decade till the first nucleotide sequence was found. The fact is kind of boring, but I found it's very interesting because we can know more about the history of the of what we have of what we are learning now, and we still need to know more about uh, the who go first, who go before us to like recognize their contribute. Yeah, it's kind of go abstract, but I think I love to sharing this. I think I need to share this to everyone who watching this. So uh, let's get back to what we have stopped. To where we have stopped. So the mass spectrometry method is, is defined as the process of assigning a name to a protein interest. We can go short at POI, and we can go short to a POI beyond its amino acid sequencing. Typically, only part of the protein sequence need to be determined experimentally in order to identify the protein with reference to the databases of the protein sequences deduced from the DNA sequence of the genes. Further, the protein characterization may include confirmation of the actual N minus and C minus termini of the protein of interest, determination of sequence variants, and identification of any post-translational modifications present. And there are some methods using the mass spectrometry. We can see it now. There are three. Yeah, there are three methods that I have just listed above. That's it. An N minus and C minus harmony, the post-translational modification, and the whole mass determination. So, oh, it has go to our limited of time, so we have to go to the end of my presentation in the short term. I just feel kind of sorry because uh, till the end, I still cannot answer my own concern, but just the boring theory I have listed above. But one way or another, I hope it will help you more or less in your own future development. Who knows? Maybe one day you have your own laboratory and you can test all those theory I have listed. By the way, this is the reference list. Is where I get my knowledge in the in this little research. Yeah, I can call it that. So thank you for your listening, and that is the end of my presentation. I wish you a good weekend. Keep safe and play cool. Goodbye.